Hi, everyone. Thank you so much for joining us here at Taste of Science. It is a wonderful Saturday in Houston, and we are so excited to be able to present Brian Kirz. Um, Brian, welcome. Just a quick introduction to Brian before we get into the swing of things. Brian is a computational biologist at the University of Rochester. He works in Nancy Chen's lab. Computational biology itself is the focus of today, so we will get into the specifics of what Brian does later on. But before we get into that, I wanted to just get to know a little bit more about you, Brian. Can you tell us how you got into computational biology?
Sure, absolutely. And first of all, thanks so much for inviting me. I wish the weather here in Rochester was as nice as Houston. It's been freezing cold all week, so I'm a little bit jealous of you. The way I got into computational biology, well, I'm first of all, I'm a second year grad student in Nancy Chen's lab. I majored in computational biology in an undergrad. But before then, all the way back when I was in high school, I had heard that Jennifer Doudna, the scientist who would later win the Nobel Prize for discovering CRISPR, the new gene editing technology, was giving a talk at UCSF, which is close to where I grew up. And I had heard about her from science podcasts and reading, and I was very excited to be able to see her sort of in her natural academic habitat. So I got permission to skip school. I skipped school with permission. Like my parents let me go. And I went to this seminar that she was giving about her new technology at UCSF. And, I was sitting there, a young high school student in a room. I think it was mostly med students there who were thinking about all the ways this technology might transform the world, which it sort already has begun to. And one of the things that she said that really stuck with me was, she looked out in the audience, large crowd of young people, and said, the jobs that you will be doing don't exist today. And so for me, that was sort of when I knew that was what I wanted to pursue. It was a really fun way to marry a lot of different interests that I had. So computer science and biology, putting these things together. I've always been super interested in evolution in the natural world, but I also like the sort of much more mechanical puzzles that computational problems can solve. And so I saw that there was a really, really cool new niche opening up for people who could apply both of those things together and answer questions about the natural world. And so then I went to computational biology in undergrad, where I sort of began my research journey. I didn't even think I was going to go into grad school until my final year. Much later, I just sort of assumed that I was going to go into industry or tech because I was interested in the applications of CRISPR. But I was introduced to sort of an academic alternate path by a mentor of mine in undergrad, who encouraged me to keep going. And I did my research. I applied in Nancy Chen's lab. We can talk a little bit about why. And I ended up here, and I'm absolutely loving that this is my job, and this is what I get to do all day, and ponder these questions, and use these tools to discover things that no one has seen before.
100%. So before we get into the next question a bit deeper here, you saw at the bottom there was a ticker that gave my best effort at defining computational biology. But just so we are all on the same level, how would you describe your work to me, not a scientist?
Sure. So, nowadays, especially, the field of genetics is moving closer to a lot of different fields. So for a very long time, the sort of minute, the biological questions, the sort of thing you could answer under a microscope, the evolutionary theory questions, right? The thinkers, like Darwin and Fisher, who were much more theoretical. And the computer science people were sort of all very separate in dealing with really different problems, right? The computer science people were trying to understand, okay, what's the most efficient way to decrypt something or align something or search something. And for a long time, these different fields were very separate and focused on different questions. And I think over time, and I'll talk about this a little bit in my presentation, there have been many different technological and theoretical advancements that have sort of brought them together. We now have the computing power, we now have the raw sequencing data to take these massive, massive amounts of information, just raw information, and process them. And it turns out that a lot of over and over again, sort of throughout the history of population genetics and evolutionary biology and computational biology, we found that the theories that we have, the methods that we have, just are not enough to swallow like the hose of information that is being produced by what our technology can give us. So what computational biology is, and what the work I do is figuring out ways to take these massive amounts of genetic data, process them, digest them, make them understandable with these using computational tools, and then on the other end, turn it into something where we can draw conclusions from these sort of massive data sets.
Absolutely. So I guess that leads me very naturally to my next question, which is one of the greatest tools that I think you probably have today at your fingertips is AI. It's a tool that lets you hopefully work with a tremendous amount of information and make it easier to consume and to make sense of. This is something that was pretty new and aligns with the high school talk that you were discussing. That couldn't have really been predicted 10 years ago. How is AI influencing the way that you do your work, and how do you think it's making or changing your field?
That's a great question. I mean, I think there are multiple parts to this. Obviously, right, we've seen huge breakthroughs in AI in recent years, and it's not, I think, adopted as widespread in the field as I think it possibly could be. But there are certain key areas where AI has already shown huge improvements in what we're able to do. So, for example, AlphaZero has solved or is much closer than we ever thought was possible to solving protein folding,
Right.
which is a classic computational biology problem, right? You have these extremely long strings that can be taken from the genetic code that turn into the proteins that make up our bodies, and for all sorts of medicine and pharmacy applications. You would want to predict, okay, how does this protein fold? How is it going to interact with other molecules, potentially with medicines? And AlphaZero has solved that using AI. So it's an extremely mathematically difficult problem that people have spent 30 years trying to figure out. But it turns out that if you build a model right and you just shovel this massive amount of data and information into it and put that in the training data, you can start to really understand super well how these proteins will come out folded. And so this is going to, over the next 10 years, I think we're going to see huge leaps forward in medicine that are directly can be contributed to this question. But this is a really specific problem, right? You have a sequence and you want to know what it folds into. The rules that you are trying to infer, which is what AI is good for, right? Those are super complex and sort of beyond the scope of humans to be able to understand clearly. There are other things that I think are more difficult. So, for example, in my subfield, when you talk more about population genetics, I've already seen some applications of AI that have started to sort of breach into the field in terms of like demographic inference, ancestral inference, making predictions, right? But first of all, you need a lot of data that it can be treated the same way. And you need replicable results. And I think that still a lot of scientists, I think most of the field of population genetics is still getting used to kind of the amount of data that we have. So they haven't quite caught on yet. I still see a lot of trepidation when it comes towards using tools that are difficult to replicate, right? For example, with chatbots, with things like ChatGPT, where you pass a test by answering certain questions, there's room for error. It's a lot more qualitative. But when it comes to when you really want to be quantitatively precise, I think those are the areas in which AI is most useful. So to answer your question about AI, I think it has been adopted in certain areas for specific questions. I think those are the sort of the most low-hanging fruit and the ripest areas for improvement. But a lot of the more qualitative stuff, I think what AI hasn't been able to do yet, that I think that collaboration with different fields of science will bring you, that's the only thing that can bring you this right now, is asking the right questions, is having the background context to understand what is interesting about this, what jumps out at me. I think that requires a lot more long-term deep study to not end up recapitulating the same mistakes or taking for granted the same assumptions that people have had in the past. So I think there's all sorts of potential. Has not happened yet, but it's not like the whole field is going to disappear because of AI. It's a really useful tool.
Right. And I think it's a sentiment that I've heard echoed elsewhere. It's that it's going to be a really powerful tool, but humans and people still have to come up with the questions and the best way to kind of leverage AI itself. Thank you, Brian. That was a great start. I want to give you some time to get into your research and tell us about what you do on a regular basis. Let's get into the slides.
Sure, absolutely. I will add this to the stage. Can you see that?
Yep.
Yeah, so I work here. This is a much prettier view than you normally see this time in the winter. It's freezing cold out. It's all covered in snow. The river's frozen over. I work in the building on the right in the University of Rochester's biology department. When people ask me what I do, what my PhD is in, it's technically I would say evolutionary biology, but it's a lot more than that. And I can use all the computational tools that I learned in undergrad because of what I do in my lab. So the specific name for the program that I'm in is referred to as E2G2, evolution, ecology, genetics, and genomics. I think especially with genetics and genomics, that can be a confusing distinction for a lot of people. So what I wanted to do is walk through the definition of what the title is, so people can get a better idea of what the background is. So evolution is the process of change in populations over time through natural selection and other mechanisms, right? So think of Darwin and the finches, think of the common ancestor of humans and chimpanzees, for example, slowly over time through descent with modification, changing their form through natural selection. Now, many people will define it in different ways. So it's true that evolution occurs as the process of genetic change, but back in Darwin's time in the late 1800s, there was no idea of genetics. Darwin thought that inheritance was passed by little tiny things called gemmules, which you can basically think of as like inheritance particles, right? This is all theoretical until 70 years ago. Ecology, on the other hand, would have been much more familiar to Darwin. That's the study of interactions between organisms and their environment. From genes to ecosystems. For a long time, these things were separate, and nowadays we're realizing that they play more of a role and have more interaction than we thought. So this is the kind of thing that you don't need a microscope. Well, unless you're studying bacteria, you don't need a microscope to study. You can go out into the field, make observations, understand how animals interact with each other and the environment around them. This is really important for me as a computational biologist. It's one of the tools in my toolkit, because a lot of times, as I was saying with AI, you have to ask the right questions, and the same result that I would get out of a genetic analysis means something completely different in the light of a different ecology. So understanding how an organism behaves in the real world, if you want to understand how it evolved, its evolution, its relationship with its environment, you really need to know the ecology, right? I work in a dry lab, which means I don't wear a lab coat and safety goggles 90% of the time because I mostly work with computers. But I work around a lot of people, and I'm constantly studying things about my study system and other study systems out in the wild to understand where to go next, right? I think this is one of the ripest areas for people to find opportunities to ask the right questions. Genetics is the study of individual genes, inheritance patterns, and gene function. So this was sort of the only thing that was possible for a long time. From the discovery of the DNA molecule in the 1950s, for a long time, right? You could only isolate individual genes. A lot of the theory only worked when you're looking at a single locus or sort of location on the genome, and how those were passed along. This is sort of what people classically think of when they think of genetics. Genomics is a little bit different, right? This is sort of more of a bird's eye view of the genome itself. So genomics is the analysis of an entire genome and how genes work together as a system. This was not possible until the methods that were developed by computational biologists opened the door to sequencing the entirety of the human genome. So in 1990, the Department of Energy and the National Institute of Health got together to do the Human Genome Project, where they were going to sequence the entire human genome end to end. And there was a great big race to this. They poured in an enormous amount of money. I think it was like $3 billion. And then in 2000, 2003, early 2000s, they finished. They finished almost all of the human genome, and that really, for me, is it's an incredible step forward, but this is what unlocked the door for what I do, looking at an entire genome, how different parts of it interact, and in my lab, in a population genetics lab, looking at how the different genomes, not just within a single individual, interact, but across an entire population. Right. This is exactly what I was talking about earlier in terms of taking in massive amounts of genetic data and trying to extract conclusions from it. And now sort of I want to transition from this introductory part where I talk about the title to some basic genetic stuff to get everybody caught up to speed, so we have a little bit of background for trying to explain a little bit more about the work that I do. So very, very simply, I'm going to do a one slide review of what you would take in your sort of college intro genetics class. Our genetic code is stored in a molecule in every single one of our cells, identical or almost identical, called the genetic code. It's stored in a molecule called DNA, and this holds the instructions for how to make any organism, humans, bacteria, every living thing on Earth comes from the same genetic code. The nucleotides that are each of those little points in the DNA connect with each other, and if read in groups of 3, become proteins, right? So each corresponds to an amino acid. Those amino acids get put into a long chain. Those chains fold, like and alpha 0 was able to predict how, and those things build up to become an organism. And through the process of natural selection, DNA has gotten very, very, very good at replicating itself. The central dogma, all you really need to know, is that our genetic code gets turned into RNA, which then gets turned into proteins. So it's sort of a down the chain thing. For a long it's called the central dogma. I mean, originally it was coined sort of ironically because it's way too simplistic a model. And of course, as with everything, the more you learn, the more you realize that there are exceptions to the rule. But very, very simply, our genetic code translates into amino acids which create proteins and which creates, the diversity of life that we see on Earth. What I do, and what I'm currently doing, is assembling a genome. And so this is, for a long time, this was not really possible because we didn't have the technology that was able to do it. And recent breakthroughs have really helped with that. So the first thing you do when you want to assemble a genome is you can't go out and find an organism and pick and go through this long ribbon and read every single nucleotide on it. You have to break it up into pieces and then put it back together. So the first thing you do is you make a ton of copies of whatever genome you're looking at, and then you put it through a shredder. Basically, imagine taking this morning's copy of the New York Times and just putting it through a paper shredder and trying to put those pieces back together, because it can only be read in those tiny, tiny, tiny chunks. We don't have a machine that would be able to read the New York Times cover to cover. We can only read, 1 or 2 or 3 words at a time. Now, the number of words or number of nucleotides that you can read at the same time has increased over the years, and that's enabled all sorts of new things. But this process is, very hard and very expensive. And where the computational biology comes in, the most important part, this is what it was basically invented for, is taking this giant jumble of shredded paper, these shredded things, and putting them back together, right? So if this piece overlaps with this piece, maybe this goes here. If this piece overlaps with this piece, right? It's a giant jigsaw puzzle that you can use algorithms to put together in a mathematically optimal way. Now, you've heard of Moore's Law, I imagine?
Yes, it's basically saying that this relates to computing power over a certain period of time. There's the computing power increases at a specific rate. I don't recall the specifics.
Right. So starting some guy named Moore, right, it was noticed during the acceleration of development of computing technology that it seemed like I think it was 2 years, every 2 years, the space that it took to, output the same amount of computing power, the size of the transistor got halved in size, right? So computers used to be so large they filled up a room, and now they can, you can fit more computing power than the entire Apollo space program inside your phone.
Right.
And so that's Moore's Law. That's exponential, increases in efficiency. What this graph shows is the cost of sequencing as compared to Moore's Law. It used to be, right? So if you think back to 2001 and the Human Genome Project, one human genome cost $3 billion to make. That's $1 per base pair. Today, I can go get my my whole genome sequence at a pretty high level of quality for about $600. It's a huge, huge, huge drop on cost. And what that means is now we have more data than we would ever know what to do with. And that's been brought about by new kinds of sequencing technology. So it used to be, you do Sanger sequencing. This was very painstaking. I hear horror stories about what grad students used to do in those times. Very slow. Then you get your second generation technology. This is what most genomes are done with. And then you get your third generation, your long reads. And I can talk a little bit about that at the end. If we have time, I want to move through this. But this is a huge, huge increase in efficiency. And what that allows us to do is ask questions that we've never asked before. It used to be, it would be too costly to sequence the genome of one tiger. Well, what kinds of questions can you ask when now you can sequence, 500 tigers spread around the world on all these different populations, understanding how they interact. And that's a lot of what I do. And now I wanted to sort of turn it over to the audience to ask people, what they think, what their sort of intuitions are over this thing. So if you paid very close attention, I did let slip the size of the human genome, so that might help you out. But just generally, if we think, a single base pair is, one little piece, 3 base pairs make an amino acid, hundreds of amino acids make a protein. How big do we think? How many base pairs long do we think that the genomes of animals are generally? I'll wait a little bit to see if there are any responses in the chat. Nadav, do you have any off the cuff guesses?
I feel like I should get the what is it? The game show soundtrack to help.
Yeah, the Jeopardy soundtrack.
Yeah, exactly. Guesses. Something tells me that the size of the animal doesn't actually correlate to the size of the genome.
Do you think it's just random?
I don't think it's random. I think, if blue whale and E. coli in the options is throwing me off, I'll admit.
Yeah. What about ranking them? Biggest or smallest out of these 4?
Now I have to rank which one. I mean, if I had to,
general guess, which one has the largest? I'll answer the question in like 10 seconds.
Yeah. I'm gonna go. I'll go with the field mouse.
Okay. It's closest to what I study, and I study a pretty big genome. Anyway, yeah. I think the answers are on the next slide.
Okay, let's see.
Right. So as you can see, GB here stands for gigabases, and so if you in the chat guessed lungfish, you would be correct. I think the answer in the top right for field mouse might be a little bit obscured. It's about 2.7 to 3 gigabases long. Giga just means billion. So this is how many billion base pairs it takes to make a genome of this size. And as you can see, there's a huge amount of variation. So this lungfish is 130 billion base pairs. And so as you can see, there's not that much of a correlation between the size of the animal and the size of its genome. And this tells us something about the nature of genome sizes, which I have gotten into completely by accident, and I'll tell that story in a bit. So this is sort of it's a dense graph, but what this is basically showing is the genome size variation major taxonomic groups. And so Nadav, you're sort of right when you noticed a little bit of a correlation there. Bacteria, you can see, are pretty darn small. And mammals, what we tend to think of as like more complex species, are much larger. The largest genomes in the world belong to plants. I think the California redwood has something like, it's ridiculous. It's hundreds of gigabases of genome. And a lot of that, I'll get into why that is. But as you can see, between taxonomic groups, there's a huge wide range. And birds and mammals comparatively, as sort of very young clades, have a pretty restricted size. We're typically between 1 and maybe 10 gigabases, and humans have a 3 billion base pair sequence. So sort of what's in all that, right? We tend to think of ourselves as more complex than bacteria. or more complex than plants, but we can see that plants have these massive genomes. And so what's going on? I have another question for you, which is what percentage of your DNA is identical to every other human? So now that we know that there's this, huge billions and billions of base pairs that make up who we are, what do you think is actually different?
Brian, can you read off the answers there? It's obscured by the.
Oh yeah, sure. My bad. I didn't see that. That's probably blocked. A is 65%, 100%. C is 85%, and D is 99%.
Okay.
So I'm sure that people have heard that we're all pretty similar, right? We're genetically very similar to chimps, but like at the base pair level, does anyone have any guesses as to A, B, C, or D? So if you took my genome and your genome and lined them up, I'll give it about 10 seconds.
Yeah, and
off the top of your head now?
Yeah, I think 85% looks promising.
Okay.
Because what makes us different? 99% could be reasonable if it's one small variation can cause a dramatic difference. I remember a while back, like bananas have like what is it? More genetic material than the average human does, or something like that.
Jeffrey and Ariel in the chat and Orlan all got it right. It's D. It's 99%. I think you can strike 100% off the list immediately because there are genetic differences between people, right? That's what causes a lot of the variation that you see: black hair, brown hair, blue eyes. We're not all exactly the same, but 99%. We share the vast majority of all these base pairs. But when you think about how much of that is in genes, not all of our DNA is directly contributing to the differences between us. So another question I have that's actually I don't think it's on the slides, but is how much of the human genome do you think is made up of genes? Like any I'm looking for any numbers, right? From 0 to 100%. How much of the actual human genome, those billion base pairs, are actually things that turn into, like I showed in the central dogma piece, DNA to RNA to protein?
Well, you would think that it would be a significant amount of it.
Right, because then what's the rest of it doing?
Yeah. I'm smelling another 99% number somewhere there.
It's actually below 5%. It's really low. It's like 3%. So when this was first discovered, when we were looking at the human genome for the first time in the early 2000s, we were completely blown away. We're like, wait a minute, what is this stuff, right? We have miles and miles of just A, T, G, just long, very low complexity regions. And most of the genome is like that. And not just in humans, in many animals. So if you think about lungfish and you think about humans, right, 130 gigabases to 3 gigabases, we seem to operate fine. The Japanese pufferfish has about an eighth of the genome size that we have, but they have the same number of genes. So there's all this stuff, just this stuff. We don't really know what it is in these intragenic regions that is contributing to these massive differences in genome size, even if the proteins that all life on Earth uses to build itself are pretty close to each other in terms of sort of raw number and size. And so I became interested in this question in my work with my study species, which is the North American red squirrel. So I work in a population genetics lab. In that lab, what we do is we answer questions about an organism's contemporary evolution and ecology through the lens of population genetics. We take sequences from a bunch of different animals over periods of time, which is kind of rare in empirical fields. And then we answer questions about their demographic history. The main study system of my lab is the Florida scrub jay, which is an endangered species, has been inbreeding more and more in recent years as its habitat has shrunk and its population numbers have stagnated. We want to answer questions about these creatures and their history. And so when I was breaking into this very interesting North American red squirrel study system, and I can talk later about why I picked that if people are interested, I wanted to make a reference genome. I am assembling right now the very first genome of the North American red squirrel. It doesn't get named after me. It's been studied for a very long time. I'm just putting it together, doing what those people did in the Human Genome Project in the 90s. I'm doing right now by myself for the red squirrel. Has never been genetically investigated before. And I found something really, really, really weird. I was coming into this from a population perspective, right? I wanted to get a reference genome so I can start sequencing lots of different squirrels and comparing them and looking at the differences. And what I found, my advisor told me was a mistake. It appeared as though, and so you can see in pink, this is kind of a complex figure. This is taken straight from one of the papers I have that I'm cooking up. But in pink is the size of the genome that I've estimated using multiple different methods. K-mers are the colored ones, and just regular assemblies are the black ones. And as you can see, for other squirrels, on the bottom there, mine's fourth from the right, it's T. hudsonicus, Tamiasirus hudsonicus. It's 6.15 gigabases. So all the other squirrels have the size of the genome that I'm expecting to find is between 2 and 3. And my computer keeps spinning out something that's 6. And I'm going, what is wrong? What am I doing wrong? So I go back and I try to fix it, and I go over and over again. And it turns out I start to believe this. And I test all sorts of different things, and we think we found something. And I'll talk about this when I publish, and I won't go into too much detail here. But even though the squirrels are very, very closely related, this and the gray squirrel that you would see on the street are about as close to each other as a human is with an orangutan. So relatively, 17 million years is not that much in evolutionary time. There has been a huge, huge shift in the size of the genome itself. And so this is what I've been investigating. This is what I do all day, is trying to get to the bottom of what's going on here. Like, what is actually making up all this stuff that is in the red squirrel genome, but is not in the gray squirrel genome, right? It's double the size, and it's still a squirrel. All the proteins that make it a squirrel are still there. Something else must be going on. And so we go back, and so I went back to the literature on this junk DNA part. I want to know what is going on in those spaces in between the genes. And what I ended up finding is that this whole wild west of things that scientists for a long time thought were completely useless, completely junk, completely non-functional, are actually very, very much alive. And I ran into something. You might hear about this if you look into a lot of junk DNA. Repetitive elements, transposons, selfish genetic elements, things that replicate themselves. And so in much the same way that a virus will replicate in a human body, or natural selection will have an animal proliferate across the planet, there are small pieces of information inside of genomes themselves that are viral in the sense that they act selfishly and they replicate to the detriment of their host. And each generation will tend to replicate themselves. So as 1 year you might have this sequence in one place, it might copy and paste itself to another place, and another, and another, and another. And you can imagine that if this gets out of hand, you can have a genome that's so big that your organism literally can't grow. It doesn't have the resources to grow. It's not infinitely. You can't have an infinitely large genome size. And so the organism, the genome itself, right, has evolved regulatory mechanisms to restrain this action. There are selfish elements within the genome itself that are trying to replicate to the expense of the organism. And the organism is trying to keep everything together, keep the revolt suppressed, and make sure that there are nice and orderly areas. And so it's this arms race between these repetitive elements that are trying to get ahead and replicate themselves, and the genome itself, which is innovating new regulatory mechanisms to make sure that they don't interrupt something functional. Imagine if you were coding, Nadav, and all of a sudden in your code there's like a comment that just like pastes itself into random spots in your code, right? You might put a comment right in the middle of a function, right? Break something super important. That's not good for the organism as a whole. And so there's a sort of power play between repetitive elements and the regulatory mechanisms that bind them. And I think I've stumbled on one where the regulatory mechanisms are currently in the phase where they've taken a step ahead. So here,
Brian,
on the left. Go ahead, sure.
One quick question. What does the timeline look like when you're talking about these repetitive elements versus the genome? Are we talking like 100 years, 100,000 years?
We're talking like evolutionary time. I mean, some of them can spread super, super, super fast, especially in large populations. I think that there are Drosophila, which are fruit flies, the most commonly studied evolutionary organism.
Because you can get cycles.
We still have samples from like wild Drosophila from like the 30s or 40s that still don't have this repetitive element. And then in the later half of the 19th century, every single wild population of Drosophila of a certain species got totally swept with this. All of them have it now. You can't go out in the wild and find one that doesn't have this repetitive element anymore. So they can move through a population extremely quickly, and they can also take many millions of years. And so the lungfish story, the story about this large variation in genome size, it's many different things, and we're still only beginning to scratch the surface of what's going on. But one of them is this expansion of repetitive elements and an organism's tolerance to that. And so what I'm showing here in this graph, on the left, you have my species, which is the red squirrel, and everything else is sort of the other related squirrels. Most of my genome, my giant 6 gigabase genome, is just repetitive elements, selfish things that replicate themselves and grow and proliferate at the expense of the organism itself. And this graph is, I think, showing it's an underestimation of just how drastic the difference is. Because remember, as a percentage, this is more stuff, right? If the red squirrel is twice the size, 60% is like literally more than an entire human genome's worth just of what could be thought of as like parasitic DNA. And so sort of to wrap it all up, to bring it all full circle, these are just some pretty pictures of things that I've been doing in terms of putting the assembly together. There are many different ways, at many different levels, the technological advances that we've seen in computing power, in AI in the case of AlphaZero, in sequencing technology that have hugely brought down the cost. All these things come together to enable us to, with the same amount of money, answer incredibly more expansive questions with much larger amounts of data. And the challenge of a computational biologist is to understand the best ways to take those massive amounts of data and cherry pick and extract, sort of derive from them the insights that we can then use in conservation, in medicine, in our understanding of evolution itself and what the genome is doing. And so sort of at the end of the day, I would say, if there's one main takeaway that I'd give about my work as a computational biologist, is it's very interdisciplinary. It relies on many different fields, right? So I have friends who are pure geneticists, pure theorists, ecology people, evolutionary theory people, and people who work on the level of methods, right? So just developing literally the computer programs that I use. All these people are necessary to collaborate with and meet with each other to answer these questions, because we need something from each of those fields. And so I feel very lucky to be sort of at the confluence of a lot of these different areas, and it allows me to answer new questions and have a job that was impossible to do even as recently as 25 years ago. And it's exactly what Jennifer Doudna was saying that day when I was in high school, which is that the jobs that you'll have don't exist yet. So that's sort of, I think, a pretty condensed version of what I do. How am I doing on time?
You're doing well. I think now would be a great time to open it up to questions from the audience. If you have any questions, please put it in the chat, and Brian will answer them. And if not, Brian, I've got plenty of questions. So
sure.
No pressure to anyone.
I see a question in the chat. Is it true that for humans to develop a new evolutionary trait, it could take hundreds of thousands to millions of years? For example, evolving from omnivores to obligate carnivores. That's a great question, right? How long does evolution take? Well, I think if you asked a bunch of evolutionary theorists, they would actually have much different answers, because the maximum speed of evolution is a huge, what counts as evolution is a hotly debated topic. I mean, if you go back to our initial definition of evolution, which is just the process and change in populations over time through natural selection and other mechanisms, right? You could argue that, human, even like in the very recent past, right? Our ability to digest milk, for instance, the evolution of skin with lower melanin percentage in order to extract more vitamin D at higher latitudes, right? These are examples of classic selective sweeps where a mutation is beneficial in a certain environment. It doesn't take that many generations. It just depends on how large is your population, how many individuals do you have to go through, and how beneficial is the trait, which means how how much more likely is someone who carries a trait than someone who doesn't in order to reproduce. And so, the pedantic answer is it just takes a few generations, but those changes are typically infinitesimal. The kinds of evolutionary traits that we talk about when we talk about human evolution happen typically over millions, in the millions of years, measured in the millions of years. But we diverged from apes only 4 to 6 million years ago, or chimpanzees, which are our closest relatives. And within that time, right, incredible innovations have been made, and there's almost nothing like it, right, in the animal world in terms of the amount of change that's occurred. Our brains getting larger, our bipedalism, right? These are really, really significant shifts. And so typically, right, you're talking about a scale of millions of years. But in my lab, where we look at populations over time, you can even see changes in allele frequencies and the frequency of mutations. You can see evolution in real time. I like to call what I do like contemporary evolution. We're understanding how populations today are different from populations even 100 years ago. And the whole reason, the thing I initially wanted to do, the reason why I joined Nancy's lab was to work with critically endangered species, right? So when you get very small populations, it becomes tougher for natural selection to work. You get more inbred. There are more deleterious recessive mutations that cause all sorts of diseases, right? You see this with human populations, but if your whole species is, only a few, only has a few members, it's very difficult to pull those back from the brink. And so what I was interested in doing initially, and what I'm still interested in doing, is using technologies like CRISPR and the assembly stuff I've described to understand how to better pull animals back from that brink. So evolution happens at many, many different scales. It could take hundreds of thousands to millions of years, but if you really want to get technical about it, the allele frequency changes that we're seeing from generation to generation are happening all the time at the level of generations. Natural selection is all around us, even today. Okay. Other question. I'm gonna prioritize the viewer questions above the mod questions. Do we know how selfish DNA might negatively impact the organism? For instance, could it shorten their lifespan? That's a great question, right? So, why is selfish DNA bad, right? Why can't you have an infinitely large genome? So the first thing is the more problematic thing is that when selfish DNA gets out of hand, it puts itself in places where it doesn't belong. So if it inserts itself into a gene that, is, your function, right, in this computer code analogy that I was using, it will break it, right? So maybe you're no longer able to, your eyes don't develop properly, or, some important part of your metabolism is broken, right? It just gets in the wrong places, and it can be problematic for that. But at the level of the genome size itself, one of the things that you notice if you go back to if I go back to the slide with the different sizes, is that bacteria have really, really low, really small genome sizes. They're mostly coding DNA. Viruses even less, right? So, and the way I would sort of analogize this is they have to run a tight ship, right? The difference between the size of the DNA molecule and the bacteria itself is a lot smaller than you or I, or an elephant, or a blue whale, or a redwood tree. One of the most important fitness traits of a bacteria is how fast can you replicate. And if you have a really large genome that's full of lots of junk, it's slower. You literally have to wait in order to copy all that DNA twice, right? Imagine trying to copy a book by hand. The book is 2 pages versus a book that's 100 pages, right? It takes a lot more time. So at our scale, at a blue whale scale, it doesn't really matter because we don't have to replicate ourselves that quickly. We don't have to grow very fast. But for bacteria, they really have to go as fast as possible. There has to be no fat. And so the selective pressures on them to have a really slimmed down, really, really tight, compact genome is really, really important. But as the animal gets over a certain size, right, you can see fungi, protist plants, when they're a little bit bigger and they're not replicating as often, it becomes less of an issue. And so there are 2 ways, but really at our scale, the only way is like interrupting functional elements. The mods would like to know why red squirrels. That's a great question. Why red squirrels? So there is a population of North American red squirrels in Kluane National Park, which is in It's in the Yukon in Canada. It's sort of between Alaska and British Columbia. And the ecology of this particular population is quite interesting. They primarily, it's sort of a taiga, very, very cold, dense forest habitat. And their primary food source in this area is the cone of the white spruce, Pinea glauca. They during the winters, right, they'll gather all these cones and the seeds from the cones. They'll make these massive piles of them called middens. They sort of live under them. But what's peculiar about this particular forest is that they are in what's called a masting cycle. So in the same way, for example, coral will breed, where they'll all explode with seeds and pine cones at the same time in a coordinated fashion. Every like 3 to 5 years, there's a massive explosion of these pine cones that happen in the forest. And so there's this huge boon. It's a feast for all these squirrels. And now the squirrels can tolerate a much bigger population. And so if you sort of look at a graph over time of the seed count in the population, you'll see these huge spikes every couple years, right? And then a drop when it goes back down to normal, and then a huge spike again. And what you see with the squirrel population size is that they increase a ton because now they have all this food, and then they starve. And they increase a ton, and then they starve, right? So they have many more children, right, in these mass years. And these squirrels have been studied for almost 50 years now, right? In the 1980s, there was a group of scientists who decided to sort of plant their flag there and every single year understand who's the father of who. They have a whole pedigree. How does inheritance happen? How many offspring did this individual have over many years? And not just that, they took DNA samples, which back then would have been totally unfeasible to sequence because the cost was just too high. And even today, it's pretty high. But now those samples have been sitting in freezers for years and years. And now it's finally cheap enough to start talking about, hey, let's go look at these squirrels. And so the reason that I'm interested in the red squirrels and what I'm going to do my thesis on is understanding, this is a really powerful opportunity. We have 4 different, there's one more part of the story that I haven't mentioned yet. It involves a lot of peanut butter. So they wanted to understand, right? So now we have all these samples from these squirrels over time. That's not very unusual in biology, right? Sometimes you have samples from a population long ago and today, right? We can make comparisons. This is true of human DNA. It's interesting stuff. What's super cool is that in 2 of the populations, starting around 2003, every year, so actually including mass years, they would ship out 25 metric tons of peanut butter and just dump it in the forest, right? In order to stimulate the conditions of unlimited food access. And so you have these squirrels that are literally gorging themselves on this food for every single year. So these squirrels have sort of they've evolved in this environment where you have these very regular fluctuations. And so they're very finely tuned to this. But now we're, every generation, there might be a different pressure. But now we turn to this experiment where we have artificially kept the food supply super high all the time, right? How does that change the evolutionary pressures that are on them? Can we see any selection? Can we see any change in allele frequencies, right? How fast does evolution occur? Is this possible to see? And so because they've sequenced all of these squirrels at all these different times, and they've separated these populations into a control group where you have this fluctuating selection, and another group where you have this consistent peanut butter driven feast, we can see what is the genetic effect of fluctuating selection versus directional selection towards a certain optimum. And how does that express itself in the genome? What are the patterns of diversity that we expect to see? How fast do these allele frequencies change? This is a really unique opportunity that is almost unheard of. I think there was a paper last year about Drosophila, which is wild flies. But until now, you've only been able to do this in the lab with flies or on computers. No one has ever had a system that's this perfect. And I feel really lucky that I'm the first person to investigate it. Okay, Jeffrey asks, how do you have files in your computer that each have the total DNA of individual squirrels? How big are those files? Great question. They're too big for the computing cluster people. They're very annoyed at me. They have to up my space all the time. What's very convenient is that, in fact, the number of gigabases that a genome is in its rawest form is very similar to the number of gigabytes that it is. So my entire squirrel genome, which I have on my computer right now, actually, is between it's about 5 5 to 6 gigabytes or gigabytes large. So they're pretty hefty, right? You can't really hold like a whole population of individuals on your computer very easily. And it's not the kind of thing that you would be able to, scroll all the way through. But each chromosome, right? I'm looking right now. They're, measured in hundreds of megabytes. But yeah, so they're pretty big, but they're like what Elden Ring, like large video games, right? Is maybe 100 gigabytes. That's like, 20, a little bit less than that scrolls.
There's something poetic
pretty big
about
files.
the translation of the actual DNA to it being stored size-wise on your computer.
Yeah.
Amazing.
Yeah.
There's some appreciation for the gaming reference, Brian.
Yeah, thanks. I put in context for people.
I've got a question for you that I think is a little less evolutionary focused, but more broadly science focused.
Sure.
I get the sense that evolutionary biology is something that people like to kind of nonsense about very frequently on the internet because it's
as it applies to like humans today. Yeah.
And you can make just like these crazy claims, like the color red is like looks good on people because it reminds people of fruit. And if you can make these claims on a website and the whole world sees it, and it's difficult to kind of trace this to any sort of reliable source. In a world where anyone can kind of post anything about this kind of stuff, what do you as a scientist feel is the best way to combat this kind of nonsense or misinformation?
Man, that's a really cutting question, Adolf. Thank you for that. I have some pretty strong opinions on this. Although I think that many people would disagree with me. What you said is totally right. I think that a lot of times sort of evolutionary ideas and especially when it comes to sort of like human psychology in particular, for some reason is very people like to root their arguments in science because it thinks that it makes them more authoritative.
Right.
Typically if you talk to evolutionary biologists or people who study human genetics, they will laugh because the amount of really clear signals of selection that we can look at in the human genome recently is not that much, right? So it used to be back in Darwin's day, right? Or when we were getting, after that, when we were getting our very first genome sequences back, we were expecting to see a lot more selection than we did. We were expecting that everything was changing all the time and that, this particular, jaw shape was like maybe better than this one. And so we would see changes over time. We found is it's almost everything is neutral, right? So that's another thing I didn't mention, but like almost, most mutations, most changes have absolutely no effect, no measurable differential effect on whether one individual or another is more likely or less to have offspring. And so I think evolutionary the ways in which this gets applied, right? Selection is not as wide, for those things is not as widespread as people tend to think. What should we do about it, right? I think people have a lot of different opinions. My personal view is that I think scientists have a responsibility to engage with the public, to go into places where the scientists themselves might not be believed or feel uncomfortable, right? I think the duty of people, the relationship between scientists and the public is super important. And that trust goes both ways, right? So I think scientists also have to, behave in ways where it's likely for the public to believe them, because when the public sort of loses faith in the institution of academia or science, that can have really, really harmful effects. And a lot of times it comes from the way scientists are engaging. With media. So, I'll give you an example of the way that these relationships are important. My study system, these Kluane red squirrels, happen on the Champagne-Aishihik First Nations reservations in Canada, and the relationship between indigenous communities in North America, all over the world, but particularly in North America, and the sort of genetics community has been fraught for a long time, right? Non-consensual taking of samples, arguments over remains, what to do with the remains, and that kind of thing, and the result has been, because of a distrust between these populations and other marginalized populations and scientists, is that they, the Navajo, for example, had like a 20-year moratorium on anyone getting sequenced, right? So no genetics for you scientists. But what this means is these populations get left out of the databases that like 23andMe and the UK Biobank, that all of science works off of, right? And so if you have groups that are disproportionately represented, that directly translates to who is the medicine, right, that is developed from these things tailored for they're the ones who are losing out on these innovations. And so I think it's important I think it's scientists' responsibility to make sure that we not just with different groups, but like with the public as a whole. And I think that we should collectively, scientists, science communicators, and members of the public, like really take a hard look at like where is the public likely to encounter scientists? How are they likely? I would love to see more scientists doing sort of direct to people communications. A lot of people, I think this is the least hot take that I've given, is that the way that journals are set up, the way that papers are set up, right, it can make it really inaccessible for people who are interested in science but not necessarily scientists themselves to sort of stay abreast of what's going on. And to communicate. So I think accessibility is super important, and I think that scientists need to sort of actively engage with what's going on. I know some people don't like doing that, and they can stay in their ivory towers. But for in every field, I think that every field deserves some like good communicators who are out on the front lines and explaining what they think is super interesting and really cool, and hopefully bringing scientific institutions and the public closer together.
100%. You're practicing what you preach, doing this with us now.
Thank you. And I thanks so much for the opportunity. I don't often get an opportunity to do this. This is a great platform.
Yeah, we're glad to host you. I think we have time for one more question here. It's a great one. Go for it.
Why is the redwood genome so large? Oh, that's a great. Okay, this is really cool. So I'm not a plant biologist, so I don't know if the answer that I'm giving you is entirely correct, or this is why. But part of the reason why the redwood genome is so incredibly large is because it's hexaploid. A bacteria typically, right? Remember, super tight and compact. They're haploid, which means they only have one copy. Humans and most other mammals and birds and whatnot are diploid, meaning they have two copies, and one from your mom and one from your dad. And some plants, and for some reason plants are able to do this. I don't think any mammal has. This doesn't really happen often in the evolution of animals. I think it happened once in hagfish, but they can actually double their entire genome size. Every single, you have two copies of a chromosome from your mom or your dad if you have two parents. So I think redwood genomes are hexaploid, meaning that there are 6 copies of every gene that they have. So even if you divide the redwood genome by 6, you're still going to get a huge genome. And plants are great examples of animals that can, creatures, organisms that can tolerate large amounts of repetitive content. So the transposon, the jumping gene, as it's known, was discovered by Barbara McClintock in corn, in sort of agricultural corn. And part of the reason why, the corn genome is crazy, right? There are things moving around and replicating, and it's really, really the Wild West. And because you have so many offspring, I think is maybe part of the theory about it is that you have a higher tolerance for these like insanely higher mutation rates, because you have a higher tolerance for seeds that you never germinate, or there's more room for whatever reason. They're more tolerant of it. There's more room for experimentation. Whereas if you doubled the human genome size, I don't think the embryo would be viable. We're sort of, in the grand scheme of things, we're very fragile creatures from a genetic perspective.
Very, very interesting.
Any last thoughts now? Anything you wanted to ask me
Well, I might have to. I have plenty of questions for you, but I think we are almost at that time. Thank you so much, Brian, for joining us. It's been a real pleasure. This hour flew by. I do invite anyone, if you have a last-second burning question, please put it in the chat. But otherwise, Brian, thank you again. Thank you to our moderators. Thank you to everyone that came. It was an honor.
Thanks so much. I really appreciate the opportunity.
Fantastic.
All right.
All righty. Take care.
Enjoy your Houston weather.
We will. Stay warm up there. Bye-bye.
Bye-bye.